Application of plant cell culture in crop improvement. Introduction. Plant tissue culture comprises a set of in vitro techniques, methods, and strategies that are part of the group of technologies called plant biotechnology. Tissue culture has been exploited to create genetic variability from which crop plants can be improved to improve the state of health of the planted material and to increase the number of desirable germplasms available to the plant breeder. Germplasm are Germplasm are living genetic resources such as seeds or tissues that are maintained for the purpose of plant breeding, preservation, and other research uses. Tissue culture protocols are available for most crop species, although continued optimization is still required for many crops, especially cereals and woody plants. Tissue culture techniques, in combination with molecular techniques, have been successfully used to incorporate specific traits through gene transfer. In vitro techniques for the culture of protoplast, anthers, microspores, ovules, and embryos have been used to create new genetic variation in the breeding lines, often via haploid production. Haploid is the term used when a cell has half the usual number of chromosomes. Cell culture has also produced somaclonal and gametoclonal variants with crop improvement potential. Somaclonal variation is the variation seen in plants that have been produced by plant tissue culture. Chromosomal rearrangements are an important source of this variation. Somaclonal variation is the variation seen in plants that have been produced by plant tissue culture. Chromosomal rearrangements are an important source of this variation. Gametoclonal variation has been defined as the variation among plants regenerated from gametic cells in culture. The culture of single cells and meristems can be effectively used to eradicate pathogens from planting material and thereby dramatically improve the yield of established cultivators. Large scale micropropagation laboratories are providing millions of plants for the commercial, ornamental market and the cultural and the agricultural clonally propagated crop market, with selected laboratory material typically taking 1 or 2 decades to reach the commercial market through plant breeding. This technology can be expected to have an ever increasing impact on crop improvement as we approach the new decade. Applications. The applications of various tissue culture approaches to crop improvement are breeding and biotechnology, wide hybridization, haploidy, somaclonal variation, micropropagation, synthetic seed, pathogen eradication, germplasm preservation, plant breeding and biotechnology. Plant breeding can be conveniently separated into two activities: manipulating genetic variability and plant evaluation. Historically, selection of plants was made by simple harvesting the seed from those plants that performed best in the field in spite of the general lack of integration of most plant biotechnology and plant breeding programs. Field trials of transgenic plants have recently become much more common. More than 50 different plant species have already been genetically modified either by vector dependent agrobacterium or vector independent biolistic microinjection and liposome method. In almost all cases, some type of tissue culture technology has been used to recover the modified cells or tissues. In fact, tissue culture techniques have played a major role in the development of plant genetic engineering. Tissue culture will continue to play a key role in the genetic engineering process for the predictable future, predictable future, especially in efficient gene transfer and transgenic plant recovery. Wide hybridization. A critical requirement for crop improvement is the introduction of new genetic material into the cultivated lines of interest, whether via single genes through genetic engineering or multiple genes through conventional hybridization or tissue culture techniques. Haploids. Haploid plants have the gametophytic one half of the normal number of chromosomes. They are of interest to plant breeders because they allow the expression of simple recessive genetic traits or mutated recessive genes, and because genes and they are of interest to plant breeders because they allow the expression of simple recessive genetic traits or mutated recessive genes, and because doubled haploids can be used immediately as homozygous breeding lines. The efficiency in producing homozygous breeding lines via doubled in vitro produced haploids represent significant savings in both time and cost compared with other methods. Soma clonal variation. In addition to the variants or mutants, cell lines and plants obtained as a result of the application of a selective agent, the presence or absence of a mutagen. Many variants have been obtained through the tissue culture cycle itself. These somaclonal variants, which are dependent on the natural variation in a population of cells, may be genetic or epigenetic and are usually observed in the regenerated plantlets. Micropropagation. During the last 30 years, it has become possible to regenerate plantlets from explants and or callus from all types of plants. As a result, laboratory scale micropropagation protocols are available for a wide range of species, and at present, micropropagation is the widest use of plant tissue culture technology. Micropropagation, the propagation of plants by growing plantlets in tissue culture and then planting them out. Synthetic seed. Synthetic seed can be defined as the artificial encapsulation of somatic embryo, shoot, bud, or aggregates of cells or any tissue which has the ability to form a plant in vitro. The use of synthetic seeds as an important on more than, as an improvement on more traditional micropropagation protocols. In facultatively propagated crops may in the long term have a cost savings as the labor intensive step of transferring plant from in vitro to soil or field conditions may be overcome. Pathogen eradication. Crop plants, especially vegetatively propagated varieties, are generally infected with pathogens. Strawberry plants, for example, are susceptible to over 60 viruses and mycoplasms, and this often requires the yearly replacement of mother plants. The excision and culture of apical meristems, the meristem with 1 to 3 of the subjacent leaf primordia, coupled with them with therm or chemotherapy with the term with therm or chemotherapy have been successfully employed to produce virus free and generally pathogen-free material for micropropagation. Germplasm preservation. Germplasm is living tissue from which new plants can be grown. It can be a seed or another plant part, a leaf, a piece of stem, pollen, or even just a few cells that can be turned into a whole plant. Germplasm contains the information for a species' genetic makeup, a valuable natural resource of plant diversity. Thank you.